Good morning, everybody, and now welcome to session 1. So now we will talk about the importance of gamma delta T cells. It is really very well known that the alpha beta T and CD19 B cell depleted transplant is superior for clinical benefits to unmanipulated and mismatched unrelated transplantation. And this is particularly important in terms of faster immune reconstitution, lower transplanted-related mortality, and improved acute and chronic GVHD associated with improved relapse-free survival. And this is due to the finding that in the graft there are not only hematopoietic stem cells, but also mature gamma delta T and Necker cells. They may protect the host, the transplanted patients, against tumor relapse and life-threatening infection. And it has also been shown that, and well documented, that high level of gamma delta T lymphocytes after allogenic transplantation was significantly associated with a lower risk of relapse, fewer episodes of viral infection, better overall survival, improved disease-free survival, and unaltered acute GVHD incidence. In humans, there are two principal gamma delta T cell subsets that are the V delta 1 and V delta 2 cells. They are different in terms, for example, of tissue distribution, and also because they may recognize different ligands and receptor on target cells. So the V delta 1 exert dominant antiviral activities, whereas the V delta 2 subset exert principal anti-tumor functions. More importantly, both subsets may be easily or quite easily expanded both in vivo and ex vivo. And for these biological reasons, we may translate gamma delta 2 cells into the development of novel cell therapies. To date, there are three main strategies to use gamma delta T cells in cell therapy. The first is the use of unmodified or unselected gamma delta T cells. The second, as already mentioned by Professor Rupert Rettinger, is the use of engineered gamma delta T cells. And the last is the use of selected gamma delta T cell subset, and then the use of expansion using different reagents that you can use. So now you will hear three different outstanding speakers that will talk about different aspects on gamma delta T cell biology and mechanism of action, also implying importance on impact in the clinical setting. I'm going to introduce Professor Bruno Silva-Santos. He is a group leader and vice director at the Instituto de Medicina Molecular in Portugal, in Lisbon. He has been awarded with many prizes, awards, and international grants. He also organized the last International Gamma Delta T Cell Conference last year, and he published many important papers in the international journals highlighting the mechanism and importance of gamma delta T cells.
Thank you very much, Irma. Thank you very much for the introduction. It's a pity I cannot be there with you, but it's a pleasure to be able to share with you our research focused on gamma delta T cells. So I'm going to share my screen. And so I'll be telling you about our attempts to develop an immunotherapy based on gamma delta T cells, actually an adoptive cell therapy, and our initial proof of concept that led to the application of these cells for treatment of acute myeloid leukemia. These are my disclosures. Basically, as Irma just mentioned, there are two main subsets of gamma delta T cells, although there is a third one that Rupert alluded to, V delta 3s, for which we still don't know much about, and we lack protocols to expand these cells to high numbers compared to V delta 1, V delta 2s, which we can already expand to very high numbers in the lab and in the clinic. And so I'll be telling you mostly about V delta 1 T cells. My colleague Murad Mamedov will focus on V delta 2 T cells later on. Both of these subsets are very, very potent against tumor cells. As Irma mentioned, V delta 1s are also especially potent against viral infection, and this may be very useful to prevent CMV reactivation and infections. Whereas V delta 2s have an outstanding response to bacteria and also to parasites, because these microorganisms produce high amounts of the ligands for their T cell receptor for the dominant subset of V delta 2 T cells that recognize phosphoantigens that bacteria and parasites produce in very high amounts. We've recently reviewed basically what we know about these two subsets and how they react to cancer, and also their potential advantages compared not only to alpha beta T cells, but mostly to NK cells, because as you'll see, there are striking parallels between gamma deltas and NKs. And this is particularly interesting in the context of the symposium, because gamma deltas and NKs are there in the haploidentical stem cell transplant graft and can indeed be boosted for better responses. So, like Rupert has reviewed a few years ago, the potential of gamma delta T cells for all kinds of allogeneic therapies is very high, because they are not restricted by the MHC complex. Data that Irma alluded to from Lawrence Lamb and others has shown that the reconstitution post-transplantation of the gamma delta T cells really associates with better prognosis for these patients. And within the gamma delta T cells, it should be noted that the striking cells that were expanded were V delta 1s, which is one of the lines of data that has inspired us to focus on V delta 1 T cells. Now, V delta 1s are less abundant than V delta 2s. They are usually only about 10 to 20% of the gamma delta T cells in the peripheral blood. And so our breakthrough a decade ago was to devise a clinical grade method to expand V delta 1 T cells from the peripheral blood. So we start with alpha beta T cell depleted products, and we essentially expand the gamma delta T cells with a combination of TCR agonism and cytokines. And what's striking is that although we are not using an anti-V delta 1 specific antibody for this expansion, what we get via this combination is a very marked enrichment for V delta 1 T cells. And we've shown that whereas V delta 1 cells can withstand strong TCR stimulation under these conditions, V delta 2 T cells cannot thrive, and actually they undergo activation-induced cell death or exhaustion energy, which may be related to the disappointing data with the initial V delta 2 clinical trials. And so on the V delta 2 cell front, there's a lot to be done, and I'm sure Murad will dwell on it later on. Now, on the V delta 1 front, we got this protocol. As you can see, from a very minor population among leukocytes, we could get above 70% of the cells being V delta 1. The contaminants left over is being V delta 2s or NK cells, and alpha beta T cells basically inexistent. And so we got to this protocol in an unbiased way by basically combining all kinds of T or NK cell agonists in the presence of different cytokines, and the protocol is published, and so I will not dwell on it. But what we've really showed is that this cellular product after 2 weeks, now the protocol takes 2 weeks, is extremely selective in terms of just targeting tumor cells or virally infected cells, but sparing all kinds of healthy cells, not only blood cells, epithelial cells, endothelial cells. Everything that is healthy is not recognized by these so-called dot cells for delta 1 T cells. whereas everything that is transformed tends to be recognized. These are 4 primary AML samples as examples. These are 3-hour killing assays where you see the impact of the addition of the dot cells, the black bars, compared to the controls, and basically the tumor sample undergoes apoptosis very extensively within these 3 hours. By contrast, all kinds of healthy leukocyte populations are spared by the dot cells, so there is no induction of apoptosis on healthy cells. We also showed that this is not selective to any tumor type, so this is AML that I'm showing you above, but basically against all kinds of hematological or solid tumors, we could find cell lines that were very well targeted by our dot cells, and so they should be recognizing ligands that associate with a transformation or viral infection, in this case in cancer, with the transformation process. And this is not just in vitro. In vivo, we were able to show that dot cells are extremely potent, and basically in patient-derived xenografts, and we established more than 4 different models of AML, CML, and ALL, and CLL as well. So basically, we could show that whereas control animals, of course, have a very high tumor burden, animals that are treated 1 week after tumor injection, and so when the tumor is already detectable in the blood, when they are treated, they are tumor-free at later time points. The bone marrow is clear, and the other tissues where there's leukemias disseminate, not only the blood, but the liver, for instance, are also essentially tumor-free. All the preclinical data has led that our startup company that we started here in Lisbon called LymphAct with the dot cell technology was acquired first by Gamma Delta Therapeutics in London and last year by Decode Pharmaceutical, and the clinical trial using the development of dot cells, now called TACO-12, is taking place in several centers in the US, including Stanford, and so the results will be known later in the year for this phase 1 clinical trial, this dose escalation. Today, I'm going to tell you about more research on how these cells may recognize tumor cells. Now, before I go into that, into the endogenous mechanisms by which dot cells recognize tumors, let me just show you that if you transduce dot cells with CARs, these are extremely potent. And so it's no surprise that if you take an antigen that is widely tumor associated, for AML we chose CD123, you generate CAR dot cells. We have high transduction with the baboon construct that Rupert alluded to. And so what you can see is that although after 42 days, we had the dot cell treated animals completely tumor-free, and the CAR dot cells treated animals can only do as well as the dot treated animals. Basically, with or without a CAR, dot cells are able to control tumors. If we then mimic a relapse by re-injecting more tumor cells into these tumor-free mice, so the mice are not retreated, but they receive more tumors at day 47. And what you can see is that now the mice that were only treated with dot cells developed more tumor than the animals that received the CAR dot cells. So CAR dot cells are more potent than dot cells, as you would expect, but actually in terms of in vivo models and xenograft models, we can only disclose this extra activity if we re-challenge the animals and mimic some sort of a relapse in vivo. Now, interestingly, back in vitro, if we test the activity of dot cells against a panel of AML primary samples, what you can see is that the impact of dot cells, untransduced dot cells here in black, or CAR dot cells here in red, varies a lot across samples. So in this first patient sample, you can see that the first bar is the control without any gamma delta cells. If you then provide dot cells, you get a minor a reduction of the tumor cells in vitro, but the CAR dot cells are able to wipe out completely the sample. So in this scenario, for this patient sample, CAR dot cells are much better than the dot cells. By contrast, in these other patient sample, you can see that the native dot cells, untransduced, are able to kill most of the tumor cells, and therefore the CAR dot cells can only have a minor, albeit still significant, addition to this efficacy already. And the third example I give you here is somewhat intermediate phenotype. So what you can see from these kind of examples is that there is a variability between the propensity of different patient samples to be targeted by dot cells. And so an important question is, what determines the level of tumor cell killing by our dot cells? And so that is the question I'm going to address for the remainder of my talk, which has two important consequences. First, to explain how different patient samples may be more or less targeted by the native dots, so without any CAR transduction, I recall. And on the other side, to allow us to understand better how dot cells may be able to recognize selectively the tumor cells and spare all kinds of healthy tissues. So one thing, and our favorite hypothesis that I will show data for, is based on NK cell receptors. And why this? Because early on during our research, we, by phenotyping our product, we realized that not only we were expanding V delta 1 cells, typically way above 1,000-fold expansions with our protocol, but the phenotype of these V delta 1 cells was changing and becoming much more NK-like. So although V delta 1 cells express constitutively the important anti-tumor receptor NKG2D, which is this dashed line here, the ex vivo V delta 1 expression, you can see that after our protocol, the expression on the dot cells is log-fold higher. DNAM1, the same thing, although it is naturally expressed, is now much, much higher on dot cells, and some NK cell receptors that were meant to be NK cell specific, like NKp30, for instance, and are not present ex vivo on any gamma delta T cells, they are expressed de novo on a subset of these V delta 1 cells. So they now have a repertoire of high amounts of NK cell receptors, and this really allows them to be extremely cytotoxic. You can see here in this degranulation assay that if you add to the dot cells any kind of tumor cell line, KG1 or HEL AML cells, essentially 100% of the dot cells degranulate against the tumor. This is even above the positive control, which is PMI stimulation. So essentially we have super cytotoxic cells, and we think that they are essentially NK cells with a twist, the twist being their T cell receptor that allows us to expand them to very high levels, and we think is also very important for the tropism and maintenance of these cells in vivo in ways that surpass the homeostasis of NK cells. That is our working hypothesis. So, but for today, I'm going to focus just on the NKRs as mediators of this cytotoxicity. Sorry, and as you know, these NK cell receptors from NKG2D to the NCR family, DNAM1, they can really aggregate a series of mediators that are not only involved in the granulation, but also in secretion of type 1 cytokines like interferon gamma. And so this is very important for their anti-tumoral activity potentially. And so what we wanted to know was on the AML side, what counterligands could be expressed that could interact with this repertoire of NK cell receptors that our dot cells express. And by screening a large panel of AML cell lines, I mean, you can see just the summary data on the right, it was very clear that two ligands stood out. These were PVR and B7H6. So PVR, like Nectin-2, are ligands for DNAM1, and B7H6 is the ligand for NKp30. By contrast, all NKG2D ligands, from well-BPs to MICA, MICP, are essentially not upregulated compared to the baseline in all these AML cell lines. And possibly you can speculate that NKG2D surveillance is so strong that there is immune evasion in most well-established tumors that have actually downregulated the expression in leukemia. It's well shown that NKG2D exerts a very strong pressure over tumors. And so what you can see is that basically the cells that are being killed by our dot cells typically have high levels of PVR, high levels of B7H6, and moderate levels of Nectin-2. These are just correlations to establish any kind of functional role we employed CRISPR-Cas9 gene editing, and basically we generated single, double, and triple knockouts for each of these ligands. And so what you can see is in these short-term killing assays where we co-incubate dot cells with the AML cells, and we've done this gene editing in two AML cell lines with similar results. So what you can see is the control is without any, so the mother cell line without any ligand being knocked out. If you take out PVR, you lose around 40% of the reactivity from 100. This is normalized, of course, to 60%. If you take out B7H6, you lose around 30%, and they seem to be additive in the sense that taking out both PVR and B7H6 removes 60% of the reactivity. By contrast, Nectin-2 showed absolutely no impact, and so we came to the conclusion that PVR was by far the dominant DNAM1 ligand, and on the NKp30 side, there was B7H6 also contributing to this V-delta-1 dot cell reactivity against AML. Now, these cell lines are produced in parallel, in separate wells, and they are kept separately. So we really decided to establish a better model where all these cells would be in competition for targeting by dot cells. So essentially what we've done was to label the single, double, and triple knockouts with different fluorescent dyes that were titrated so that they would not cause any kind of viability loss. And so once we optimize this, we would have in the same well these different AML cells with different repertoires of ligands, and we could ask the question how much each of them would be targeted, each of these populations would be targeted by our dot cells. And again, also in this co-incubation scenario, we saw that PVR loss removed around 40% of the reactivity in this case. Nectin-2 loss did not impact at all the killing, and so the double knockout for PVR and Nectin-2 gave the same result as the PVR single knockout. So on the DNAM1 side, PVR is the key, and if you take this double knockout and then you do the triple and remove B7H6, you have the biggest decrease in reactivity we've seen so far, which is about 70% for this line that lacks both PVR and B7H6. And Nectin-2 is actually completely negligible. And this is in vitro, but when we inject these cells in vivo, we see the same thing, which is that whereas AML cells that have these ligands can be suppressed in vivo by our dot cells, the AML cells that lack these two ligands, PVR and B7H6, cannot be impacted in vivo by our dot cells. So the dot cell treatment fails in AML cells that lack these two ligands, whereas it's still able to control if they have these ligands. And by the way, this has been just one injection of dot cells 3 weeks before we assess the impact on the blood load. Why is this happening? What are these two ligands doing in these interactions between dot cells and AML cells? So to cut a long story short, as these microscopy images show you, for wild-type AML cells, we have very strong interactions between the dot cell and the AML cell. They define these so-called immune synapses that can be seen by phalloidin staining of the F-actin that accumulates at the interface between the two cells. So in red, you have these very strong synapses. And as you can see, if you have the double knockout for PVR-B7H6, these synapses become much fainter. And this is not only the eye that will tell you this, you should actually quantify, of course, and the quantification of many thousands of cells shows you that wild-type AML cells establish strong synapses, PVR knockout or B7H6 knockout, intermediate synapses with intermediate levels of F-actin at the synapse, and it's the double knockout that suffers the most. And so we really think that these two ligands are critical to stabilize this interaction that will be critical then for the release of perforin and granzymes, therefore creating the so-called cytotoxic synapse. And this is what you can see, that perforin is actually induced upon co-incubation between the dot cells with the AML cells, but this induction of perforin can also be completely reverted if you lack the strong interactions that are accounted by the PVR and B7H6 expression. So then you don't upregulate perforin above the baseline. And by the way, just because we really think this is not TCR mediated, if you block the TCR with antibodies, what you can see is that there is no impact at the synapse formation. And so this was all cell lines, and so the final slides are basically with a large quantity of primary AML samples, trying to see how these ligands behave and how much they may be informative for the dot cell mediated targeting of primary AML samples. So the first thing to note is that the expression of these ligands is much more variable in primary cells than in cell lines, as you probably would expect. So among primary cells, we have sets of samples that essentially do not express these key ligands, in particular PVR and B7H6. So this is what we call non-expressors of the key ligands, whereas thankfully the majority of the samples actually expresses one or both of these ligands. So if you segregate all the samples between those that don't express any of the ligands or that they do express one of them, what you can see is that clearly dot cells can only induce killing of the samples that express some of these ligands, whereas those that don't are basically completely inert in terms of their reactivity. And so this is the distribution of the expression of PVR, Nectin-2, and B7H6. You can see there's a very strong co-expression of PVR and Nectin-2. That will be important for the next slide. And essentially, what you can see in about B7H6 is that most of these samples actually do not express B7H6. There is very low expression of B7H6. As you can see, even the expressors that express the Genome-1 ligands are usually B7H6 negative. And so this explains why, when you segregate which ligands are the informative ones for the reactivity of dot cells, the two Genome-1 ligands are informative and they are very significantly differentially expressed between the targeted and non-targeted cells, whereas B7H6 is not. Basically, few samples express B7H6, and that expression is not segregating the reactivity of the dot cells. So these can actually give rise to the following ROC curves that are used, obviously, to see the potency of these potential biomarkers. B7H6 is not a good biomarker, like MIC-A and MIC-B also is not. And so these ROC curves really clearly show that, whereas PVR and Nectin-2 are very strongly associated, both in terms of their sensitivity, they are very good at describing a target, and with high specificity, especially PVR, it's highly specific. The fact that both PVR and Nectin-2 are now quite useful as the potential biomarkers comes precisely from the fact I showed you before, that these two molecules tend to be co-expressed. And actually, if you see in Nectin-2, there was never a single AML sample that expressed Nectin-2 without expressing PVR. So they all co-expressed PVR, as you can see here, Nectin-2, PVR co-expressing samples, which possibly explains these ROC curves for Nectin-2 is an interesting biomarker, but as I showed you before, by the functionality of the ligands, PVR is much highly expressed, and it's only PVR that when we knock out has a functional consequence for dot cell reactivity. So with this, I would conclude my talk by really summarizing these findings, that PVR and B7H6 are very high on AML cell lines and seem to be required for this cytotoxicity. This comes from their critical role at the immune synapse, stabilizing this interaction, allowing the cytotoxic molecules like perforin to be induced and released against the target. But when we go to the real world in primary AML samples, most of them actually lack B7H6 already, and so PVR is the only one that remains as a strong associated ligand and a potential biomarker. In terms of recognizing tumors versus healthy tissues, it's important to know that PVR, although a basal level is present in many healthy tissues, the high levels of PVR are only induced by these factors here: DNA damage, cellular senescence, danger-associated molecular patterns, or viral infection, making PVR an excellent sensor of transformation or viral infection. And with this, I would like to thank Takeda for the sponsored research agreement that allows us to do a lot of this research, our collaborators in our academic projects, and the people in my lab, especially Sofia Mensurado, that has done this research. And I will now take any questions. Thank you very much.
Thank you very much, Bruno, for your exciting talk and for sharing with us your data. So,
thank you, Irma.
If you have any questions, comment.
Hello, I have a question. Very, very nice talk. Can you please let us know if you perhaps checked the other components of the traditional immune synapse, like receptors like LFA1, ICAM1, which should also be expressed on both AML and gamma delta T cells?
Yes, a very good question. Actually, we did stain for LFA1, which was expressed as expected, but we have not done a thorough dissection of the synapse. And actually, we are now going into it through an unbiased approach, trying to do a proteomic analysis of the synapse. So by means now that allow you to really cross-link the proteins that are based at the synapse, and then do mass spec on all the constituents of this kind of interaction, and then having a very quantitative kind of measurement of all kinds of proteins present at the synapse. And I'm sure, of course, not only LFA1 and ICAM1 will show up, but also other molecules like CD2, hopefully our NK cell receptors as well. So one thing we have not been able to do due to the lack of reagents that are really good enough to do this is to really show how DNAM1 or NKp30 localize at the synapse in interaction with the counter ligands, and this is something we'd like to do in the future.
Thank you, Bruno. Do you have any additional questions or comments?
Bruno, this is Alice. Thank you so much, first of all, for a beautiful presentation and for being with us, even if virtually. Really appreciate that. Really impressive to see really the identification of these biomarker to predict the response. And I'm wondering if you looked at the specificity of these biomarkers on the AML in terms of DOT stimulation, or if we look at the impact, for example, of CNV on the VU-delta-1. And I am thinking about this model in vivo in a patient who, post-transplant or in a cancer immunotherapy context, and who is not receiving DOT, but he's actually having an expansion of VU-delta-1. And if the way this expansion is triggered either by AML blast itself or by CNV infection can predict the same type of behavior of the cells, or if it's just specific to the way you expand the cells?
It's a very good question, Alicia. Thank you for that. And a very tricky one, because, to really use the cells in all these kind of assays, we need to get enough cells. And, V-delta-1s are not very abundant, so we need to expand them, activate them, and so we use our protocol. So we could use other kinds of protocols, but in all kinds of circumstances, if we use any kind of TCR stimulation, we would be getting some DNAM1 upregulation. And so the levels we would have in vitro for our assays would not be the in vivo levels of DNAM1 that are present in the patient, let's say. And actually, this is an important thing, because one of the reasons that people have kind of postulated NK cells, endogenous NK cells, may not be doing enough of a job is that some of these receptors are actually downregulated during leukemia progression. And so it will be important to provide, as a DLI or as an ACT, cells that have high levels of these receptors, and that's what our dot cells have, as NK cells that have been expanded in vitro will also have. So it's very difficult to tackle the endogenous because there's no way to keep the endogenous with the endogenous phenotype and be able to basically study them in the lab. At least we have not found that. Even just IL-15 provision, which obviously is a cytokine very important for these cells, changes the phenotype of these cells. And so there's nothing neutral. The most neutral cytokine we could come up with is IL-7, but then the cells are basically alive and not doing much. And in that context, we have not assessed, in particular, they're not killing much. And so we have not assessed if DNAM1 is still relevant or not in those kind of assays.
Okay, I have just a curiosity. You mentioned that you use an escalating dose into the patients. So which is the maximum tolerated dose that you used, and at what time you infused your dot cells?
Those are excellent questions, but I will defer them to the colleagues because I'm not involved directly in the clinical study, of course. Our colleagues from Decode, and actually they are present at the meeting. So maybe as part of the discussion, there might be some discussion on that, but actually I don't know the details by heart, so I cannot tell you exactly the details of the protocol.
Okay, thank you so much. Thank you, Professor Bruno Silva-Santos.
Thank you very much.
Thank you for your excellent talk.
Thank you.
Now we can move forward to the next speaker. I'm very glad to present and to introduce Professor Alice Bertaina. She's a group leader. She's an excellent researcher and scientist and clinician. He is also co-director of the Bass Center for Childhood Cancer and Blood Diseases. I had the opportunity to collaborate with her many years ago, and for many years, when she worked in Italy, in Pavia, and later on in Rome. She was the head of the stem cell transplant unit at the Ospedale Bambino Gesù in Italy. And of note, this institution is the first institute in Europe for the number of pediatric patients transplanted with stem cells. So I'm very honored to present Alice, and thank you.
Good morning, everybody, again. It has been a great start. I have to say I'm always mesmerized by Bruno's talk, and I'm so appreciative that he could have really educated us about gamma delta T cells, and he showed this latest data that he produced in his lab. Gamma delta T cells are really a fascinating population, in my opinion, of innate-like cells, and I love their ability of really being in between innate cells and an adaptive immunity. And they have a role in the context of alpha-beta T-cell depleted stem cell transplantation, exactly because in this graft manipulation strategy, or even if we deplete extensively of alpha-beta T cells, we enrich the graft of gamma delta T cells and of NK cells. And I just report here the number of the median gamma delta T cells infused in our product at Stanford, which is about 10 million per kilogram of body weight of our patients. So we really have an adaptive infusion of donor-derived gamma delta T cells that we can use as a tool to improve leukemia-free survival, and of course also to improve protection from infection, especially in the very early phase post-transplant, where our patients are particularly immunodeficient and when the alpha-beta T cells did not recover yet. Irma mentioned that is very well known that gamma delta T cells level post-stem cell transplant, post-allogeneic stem cell transplantation are correlated with a better outcome. And so going back in time when we were in Rome, Franco, Irma, and I really collaborated to understand how we could better really leverage this population of cells in vivo into the patients. And we have run a trial in which we infused zoledronic acid in vivo into patients at serial time points. We were starting 30 days post-transplant and then continuing every month for a maximum of 5 doses. And we did a lot of correlative studies with the patients exactly to understand how the activation of these phosphoantigens through the phosphoantigen from the biphosphonate could benefit the patients. And I'm reporting this figure here in the right because to me it's the most critical in really showing why this is important. When we looked at the outcome of our patients and specifically at the patients who were alive 90 days post-transplant and disease-free versus those who passed because of an early relapse, we saw that there was this statistically significant difference in terms of activation of the Vu-delta-2 gamma-delta T cells in this case. And this was really measured by the CD107 degranulation. So our really hypothesis was those patients who are doing better are patients who have more activated Vu-delta-2 gamma-delta T cells post-transplant, and how we can leverage this in vivo and in vitro to improve patients' outcome. Fast forward at Stanford, Giulia Barbarito, who was previously working in Irma's lab, and so has been involved in our gamma-delta T cell work since the beginning, is evaluating all our alpha-beta haplo patients. And so here we report some of her data that phenotypically characterize the kinetic of immune reconstitution of the gamma-delta T cells. And you can see that very early post-transplant in the first weeks, gamma-delta T cells are the most frequent subset of T cells amongst the CD3. And this level, in terms of frequency, progressively go down, reaching a physiologic level around 6 months post-stem cell transplant. This decrease frequency of the gamma-delta T cells is paralleled by an increase of the alpha-beta T cells. However, it is pretty interesting to see how this high frequency in the very early post-transplant, it's not translated in an increased absolute number of gamma-delta T cells, which, however, remain pretty stable between day 30 and 1 year post-stem cell transplantation. So from my perspective, the main question is, okay, we don't have changes in terms of absolute numbers, but do we have cytotoxicity differences? We have molecular differences. What is there something which is making these gamma-delta T cells that we have earlier post-transplant, where the cells have a much more lymphodepleted environment to expand, that is making them more cytotoxic? And how can we learn from those cells to make all the gamma-delta T cells, either later post-transplant or for an off-the-shelf product, more cytotoxic? So to answer this first question, we went deeply in the phenotypic characterization, and here we show just the main two subset, Vu-delta-1 and Vu-delta-2. And you can see that, interestingly enough, although we infuse a graft which contain mostly Vu-delta-2 positive cells, as is expected, since most of the circulating gamma-delta T cells are Vu-delta-2, early post-transplant, day 30, we already have most of the circulating gamma-delta T cells, which are Vu-delta-1. And of course, the proportion of the Vu-delta-2 is decreased. So really, I think that the intriguing part is understanding what is driving this shift in terms of subset proportion between Vu-delta-1 and Vu-delta-2. And if we look most at the maturation of those two subsets, do we have differences in this maturation status as well? The answer is yes, because if you look, for example, on the top row, where we have the Vu-delta-1 gamma-delta T cells, you can see that most of the cells that are infused are are actually naive, while we don't have so many naive. The proportion is already reduced at day 30, and this is in the opposite direction, the same for the central memory. But for me, it was even more interesting to see the behavior of the VU-delta-2, because for the VU-delta-2, which again is the main population which is present in the peripheral blood of our adult donors, most of the VU-delta-2 cells that we infuse have an effector memory phenotype, but early 30 days post-transplant, those are mostly naive. So what is happening in these 30 days post-transplant that the infused effector memory, the gamma delta T cells we have are not the predominant population, while we already have naive VU-delta-2 gamma delta T cells. So these are really some questions that have been raised based on this data, and then now we are trying to answer with some preliminary data that we have been able to produce in the lab. So first point is, we have a patient in which, after this transplant, the proportion of gamma delta T cells is the most abundant of the CD3 very early post-transplant, but the absolute numbers remain the same, and we have some shift into the subset and maturation of those subsets. So what are the molecularly difference between those two types of cells? Are really different cells, or is just a different kinetic? To investigate this question, we designed a pilot study in which we utilize gamma delta TCR sequencing. Here you can see a schematic representation of the study design. Our idea was really to follow molecularly the donor gamma delta T cells that we infuse with the graft and their fate into the recipient at different time points. So here we have analyzed 6 patients with acute leukemias and 2 patients with non-malignant disorders. We have performed a bulk TCR sequencing in cells infused with the graft, and then we looked at the same cell into the recipients 30 days, 90 days, 6 months, and 1 year post-stem cell transplantation. So the first question that we wanted to answer was, are the clones that we are infusing with the graft the clones that are predominant into the recipients, and what is their fate later on in the evolution of the transplant? And you can see here that in at least half of the patients that we have analyzed, there is an expansion of some clones that have been infused with the graft. This is a representation of the top 10 clones that we have identified in the graft. And so in these patients, we have a temporary expansion of this clone, which usually happened in the first 3 months post-transplant, and then they decrease over time. And this decrease over time is something which actually characterizes all these patients. All the clones that we have identifying as the top 10 represented in the donor, they don't last very long into the recipients. Really, bringing us to the hypothesis that actually what we see so early post-transplant is something newly generated into the patients that can recapitulate what is the ontogeny of gamma delta T cells in a normal fetus. The next point was to evaluate the gene usages of the gamma and the delta chain. And here you have these figures which represent a specific patient. In this case, it was a patient with acute leukemia who did not relapse. And here you can pretty intuitively see that there is a big difference between the delta and the gamma chain in terms of the dominance that we can see in the delta chain. The VU-delta-1, which is this light pink part of the pie, increase over time and is the absolute dominant chain, while in the gamma chain you have an increased heterogeneity, which it's very different from what is expected in the delta chain, and also occurs later on. So we also have a very different speed of maturation of the delta and the delta chain, since we have a peak of the delta chain around 30 days, which is instead happening later around 90 days for the gamma chain. And in terms of the junction, TRDJ1 is the most used junction region for both the graft and the recipient post-transplant, while for the gamma chain is the TR-gamma J2 junction that is predominant in the recipient, while again, not surprisingly, in the graft, most of the V-delta 2 are V-gamma 9, and so this is the absolute predominant gamma chain. The next step was really focusing on those naive V-delta 2 gamma delta T cells that we see so early post-transplant. And the biological question we had about that was really, are these cells recapitulating the ontogeny of the gamma delta T cells? Is this such a lymphodepleted setting that the gamma delta T cells can behave as they behave in any normal fetus? So again, to answer this question, we need to go back and look at what's happening in a normal fetus, and is now pretty well established that in human gamma delta T cells, as in mice gamma delta T cells, the fetal development occurs in waves. And the very first wave that we see is a wave of V-gamma 9 V-delta 2 positive cells. This is the absolute first population of cells, which has a specific signature, fetal-like, and is very different from the adult-like population of V-gamma 9 V-delta 2 that we have so commonly frequent in our peripheral blood. After the 25 weeks of gestation, the V-delta 1 is actually the subset of gamma delta T cells that we see more frequently, and is going to be also the more abundant at birth. But I mentioned that fetal-like and adult-like cells have a very different signature, and in this slide we can see more in detail what differences are. Specifically, fetal-like V-gamma 9 V-delta 2 have a semi-invariant V-gamma 9 V-delta 2 TCR, which has a characteristic of phosphoantigen reactive TCRs, specifically represented by a rearrangements, which is the V-gamma 9 gamma JP. Why this is important? This is important again because we know how easy it can be to activate those cells with biphosphonate, and it is possible that those phosphoantigen reactive V-delta 2 gamma delta T cells early post-transplant are the ones that can have the better cytotoxicity. This is not the same type of TCR arrangement that the adult-like gamma 9 cells have. The second characteristic that really differentiates the fetal-like versus adult-like V-delta 2 gamma delta T cells is the CDR3 region length. Because, and here you can see that we'll see in another slide, the CDR3 length of the germline sequences, so of the fetal-like V-delta 2 is shorter, and we have clear data of those analyses in literature, while in the adult-like V-delta 2 cells, there is a high number of N insertion that is used, and so the CDR3 region becomes more variable in terms of length and heterogeneity. So we really focus in these two aspects, the gene usage and the CDR3 length, with the idea of really differentiated and understanding if the V-delta 2 naive T cells that we see early post-transplant are fetal-like or adult-like. And here are some answers to this question. So I am keeping this figure on the left just to help you with the literature data. Here we are looking at the gamma J junction and specifically at the usage of JP1. We know that JP1 is more abundant in fetal and in cord blood, as shown by this gray area here. In this case, we looked at our patients and at the V-delta 2 of these patients, and you can see that actually, consistently with what reported in literature, early post-transplant, the JP1 usage is more abundant than in the graft, and then decrease over time, exactly as it happens in the ontogeny of the gamma delta T cells. So this really corroborates our idea that the cells that we see so early are these in this light yellow and have characteristic consistent with a fetal-like signature. The same is true when we look at the delta J junction and specifically at the delta J2 gene usage, which is even more easily to understand. We can see here really that this proportion, it's much higher in the fetal blood than in the adult blood, and this is again very true for our data, where the increase of this J2 usage is especially at day 30. It peaks at day 90, again reminding us this different speed of maturation between the delta and the gamma chain. Because the delta chain we saw the peak at day 30, but the curve, the kinetic of maturation is consistent with what we see in fetal development, and therefore really consistent with our hypothesis that the gamma delta T cell reconstitution after alpha beta T cell depleted haploid transplant is recapitulating the ontogeny of the gamma delta T cells. What about the CDR3 length? I mentioned before that this is another biomarker for us to understand whether these cells have a germline sequence or an adult sequence. Here you can see these two figures are just to show you that in terms of number of N addition or CDR3 length in nucleotides, the region, the segment CDR3 is shorter in fetal gamma delta T cells than in adult, and specifically is about 45 nucleotides, which correspond to about 14 amino acid. Thus, really our hypothesis that if the CDR3 length of the gamma delta 2 associated to the V gamma 9 chain that we see at the day 30 evaluation is shorter than 14 amino acid, then we can really say that those cells are equivalent to fetal-like gamma delta T cells. And here Julia was able to create this beautiful representation that really give you a sense on how those shorter CDR3 regions, shorter than 14 amino acid, are mostly present in the sample, in the gamma delta T cells of patients before day 180, and especially really in the first 90 days. So early on, we have this wave of molecularly equivalent fetal-like gamma delta T cells that then, with the time after transplant, become more adult-like. CMV is a great expander and activator for gamma delta T cells, and is also the most frequent virus reactivation or infection that we have post-stem cell transplantation. We have previously published in our work in Rome what were the effect, especially on V delta 1 gamma delta positive T cells, and at that time we showed really that the cells of patients who had experienced CMV reactivation were then in vitro more able to kill leukemic blasts. But we also know that there are important changes at a molecular level that CMV is doing on gamma delta T cells. And so we wanted really to see how in this cohort CMV reactivation shaped the clonality of the cells that we have analyzed. And so here, what we have done was dividing the patients between those patients who didn't have a post-transplant CNV reactivation and patients who did have a post-transplant CNV reactivation, and then we look at the impact of the CNV donor serostatus. In the top row, we have a seronegative donors, and in the lower row, we have the seropositive donor. It is pretty clear how having a donor which is seropositive or seronegative doesn't impact really the clonal expansion of the gamma delta T cells. But what is impacting that is actually having a post-transplant CNV reactivation. And this is an impact which is happening both for V delta 1 and V delta 2 at different level. So we went more into details, and these 3 map plots show us the 2 patients with no malignant disorders that we have enrolled in this study. One patient experienced CNV reactivation, the other one did not. You can see really very clearly how in the patients who experience CNV reactivation, we have the expansion of specific clone, which does not occur in the patients who did not experience the CNV reactivation. And this is true for both Wu-delta-1 and Wu-delta-2, although we know that Wu-delta-1 are much more sensitive to CNV as a stimulus, and they have much more remarkable changes. But the next step really was to understand how much the CNV reactivation also was impacting the leukemia relapse. And so here there is an analysis in which we divided the patients with malignancies, those who remain in remission and those who instead relapse. This is the slide for the Wu-delta-1. The following one is evaluating the Wu-delta-2. But it is pretty intriguing, in my opinion, that all the patients who remain in remission actually experience CNV reactivation and had clear expansion of specific clone, which are responding to the CNV, while 2 of the 3 patients who did have a relapse did not experience CNV, and you can see that their profile of clonality is much more heterogeneous. And one of those 3 patients who relapsed very late, after 6 months, relapsed after clearing the CNV reactivation and losing really that expansion of clones, which, in my opinion, kept him in remission before then. The impact on Wu-delta-2 is more limited, and it's more heterogeneous, but still we see expansion of more specific clone in those patients who remain in remission and who had the CNV reactivation. So to the last question, which is also the million-dollar question, about really the presence of a specific phenotype of clonal signature that can be linked with the ability of maintaining these patients in remission. Julia did the same and looked really at the clonal expansion based on the relapse. And here you have the 3 map plots of 2 representative patients, while in this ordinary list squared of clonality across time points, there are evaluated all the patients together. So we can see that is in the patients who did not relapse, we have, again, the expansion of specific clones. And those clones are not the top 10 clone that we have seen in the graft at the very beginning. So those are new clones that are originated into the recipient and that they expand and that we think can maintain these patients in remission and can be expanded upon different drivers, different viral activations that they have, maybe even positive MRD, for example. In the relapsed patients, it's pretty clear in this type of graph how the clonal landscape is maintained highly diversified. So really, there is a signal in terms of really correlation with the expansion of some clones. We know that there is a signal of expansion of clones upon CNV reactivation. If we look together at all the patients who relapsed and did not relapse, while this remains very stable for the patients who relapse, we have a more clonal expansion in those who remain in remission. And what's going to be important next is really functionally evaluating the effect of some of these clones and understanding if the expansion of some CNV clones is also a biomarker of response to leukemia and ability to expand upon leukemia recurrence, or if it's just an impact that the CNV has on the maturation of the immune system. So to conclude, really, our data post-transplant reveal that clearly there is a different phenotype of gamma delta between the donor and the recipient post-transplant, and this happened very early. In tracking the recipient post-transplant using TCR sequencing, we have identified 10 top expanded clones who expand in more or less half of them, but quickly decreased over time, giving really space to new clones to originate. we showed how early gamma delta T cells have characteristics which are consistent with a fetal-like signature because of their gene usages and because of the CDR3 length, which is consistent with the short one typical of germline sequences. The post-transplant CMV reactivation clearly impact the gamma delta T clonality regardless the donor serostatus. And the CMV-driven gamma delta T cells clonal expansion seems to be associated with protection from leukemia recurrence, although this is, of course, something that needs to be evaluated more on a biological perspective and in a larger cohort. I really would like to thank a lot of people who contributed to this work, but first of all, really Julia Barbarito, who performed all the analysis that you have seen, David Lewis, who analyzed the data that you have seen of the TCR sequencing, all the lab members, Robbie Parkman and Ken Weinberg, for their continued mentorship, and all of you for being here and for listening to this talk. Thank you.
So 2 questions for you, Alice. The first, your initial expansion, it really feels like that's to allo clones, to allo cells, okay? Can you prove that?
You mean, when you mean allo cells, you mean derived with a graft, or?
The graft comes in, and you have an expansion. Is that to, that has to be differences between the donor and the recipient. And it would be nice to be able to prove that. Can you prove that?
So, thank you. This is an important and difficult question. So my idea was exactly that. The gamma delta T cells that we infuse with the graft homeostatically expand in this first week. We know that these cells home into the tissues, and they can just get, after engraftment or around engraftment, can circulate. However, it really seems that are different gamma delta T cells. And we know that gamma delta T cells can have an extra thymic differentiation. So the question is really, I don't think they are host gamma delta T cells. And we need to do a chimerism by using flow cytometry to really be able to differentiate between donor and recipient in this case, because they're too small proportion to use STR. And we have some antibodies that we can use for that, and we are actually looking into that. I would say that we can say that, like, some of the data that we have looked at, they really seems donor cells. They are not recipient host T cells. It's intriguing, though, the fact, again, that molecularly are changed means that some big rearrangement is happening already so early post-transplant.
Yeah. It seems like that would be something interesting to prove, and then you could target it. The second question is, the CMV relapse relationship has always really intrigued me. And what you're showing is when you have a diverse reaction to CMV, then your relapse is less.
Yeah.
In those same patients, do they have a diverse reaction to known alloantigens compared to the patients who don't have a diverse reaction to CMV? Now, the reason why I say that is this, it seems to me that you just are doing an indirect measure of, someone's ability to expand their immune system. So if you have a very vigorous immune system, and it does a very diverse and comprehensive expansion to CMV, then it's going to do that to tumor antigens too, maybe.
No, you're absolutely correct. What we see now, we can clearly say that there are clones that are expanding upon CMV reactivation. But in fact, the next question is, like you are saying, right? That means just that immune system is particularly good in responding to CMV, and we can then assume that the same immune system will be able to respond to leukemia in the same good way, or really is it CMV-specific change into the gamma delta T cells regardless the goodness of the immune system, or there is a biomarker of these gamma delta T cells in response to the CMV that we can use as a selection for a best donor, for example. And this is I don't have the answer, but that's exactly part of the fact question that this type of work is trying to answer.
Yeah, it's very interesting because if it's CMV specific and then you have a total separate thing that's preventing relapse, then that's interesting. You're just measuring a good immune system.
Yeah.
But if it's if the CMV antigen response, also those same cells attack the donor because of whatever reason,
Yeah.
Antigens that maybe are shared that are alloantigens from CMV, then CMV matters.
I would say in our prior work in proteomics, we have clearly shown that the CMV significantly impact at the proteomic level both, by the way, Wu delta 1 and Wu delta 2. We know that the Wu delta 1 is the main target for CMV, and we know that expand that more at in terms of really numbers, but in terms of proteomic changes, we saw even with the zoledronic acid that the Wu delta 1, the Wu delta 2 are very affected as well. So there is, what I'm trying to say is that obviously what we have looked at, this is just really like the tip of the iceberg. There is much more that we need to go into details, but I believe that the CMV has an actual specific change that maybe we cannot necessarily phenotypically characterize, but we can characterize at a TCR sequencing and at a proteomic level, and can give us really a tool to how to engineer those cells in those patients who are not, encountering CMV in their post-transplant life, or how can we use that in vitro to make those gamma delta T cells much more effective for everybody when we infuse them with a transplant.
Yeah.
Thank you.
Thank you, great talk. I have two questions. So the first one is that the CMV activated the clone expansion, and it's good for protection of the tumor development. So do you see the top 10 clones, whether there's common clones across the non-relapse patients?
So the top 10 clones which were in the graft are not really the same that we have seen in the recipient post-transplant. So those clones that we see expanded upon CMV reactivation and that we speculate can protect against leukemia are not shared with the graft.
Yes, but between patients.
Yes, so you're asking if the sequences that we see expanded are shared between the patients.
Yes.
They are not shared necessarily amongst the patients.
Okay. So my second question is that seems CMV activation is good for protection.
Yeah.
So can we do any manipulation after transplantation so that?
That's exactly the point. We have always known in different types of transplant setting that patients who have CMV reactivation have a better leukemia-free survival. I would say that we have never really dive deep in why biologically this is happening. I think that overall the gamma delta have a big impact, and in this transplant setting, I think that the way we can use that is exactly we want to protect our patients from CMV infection because that can be a problem, but we can mimic what the CMV is doing to the cells, and that's exactly the goal. The goal is to identifying what are the proteins, what are the clones in this case, the markers that really the CMV is triggering into these cells, and can we engineer our cells or stimulate in vitro the cells in a different way so that they can become and act as they were encountering the CMV and use them against cancer.
Exactly. Thank you very much.
Thank you.
Thank you, Alice, for your nice presentation. I have a question for you. So I was wondering if you have, for example, studied some additional viral reactivation in your patients. I mean, for example, ADIVU or ABVU may impact on your V-delta-1 clonality or not?
That's a very good question. We haven't looked at that with the clonality. We have seen that adenovirus is, which, it's actually our main problem after transplanting kids, because we have less medical therapy, and so it can be more life-threatening than CNV at this point. It is clearly mediating an expansion in the peripheral blood of both the V-delta-1 and the V-delta-2, but we haven't looked in this data about the impact on the clonality, which we will definitely do. We have all the data, so we need to just correlate them. It's going to be, again, very interesting to understand if it's CMV-specific versus virus-specific or virus-specific in that case. Thank you. Thank you very much, everyone.
Now we move with the last speaker. I don't know if he's here. Okay, thank you. I'm happy to introduce Dr. Murad Mamedov. He's a brilliant researcher who is very recently awarded with a prize for human immunology and vaccine research. He published many interesting papers also in very important journals, including Immunity and Nature. And he used the CRISPR technology to disrupt hundreds of genes in lymphoma cells and to understand which gene may impact on gamma delta T cell killing. So thank you.
Thank you for the introduction. Thank you for this opportunity to Alice and the organizers. So I'm a postdoc in Alex Marston's lab at Gladstone and UCSF, and after finishing my PhD here at Stanford in Mark Davis's lab, studying gamma deltas in malaria, I went to Alex's lab to use the tools of genome-wide CRISPR screening to understand gamma delta biology and their targeting of cancer cells. So I'm going to tell you about the story that we ended up developing during my time there. So when we think about adaptive immunity, we tend to think of cells that express somatically rearranged, diversified receptor expressing cells. So in the case of B cells, these are antibodies which tend to have high affinity for their ligands, which they bind directly without any antigen processing. In the case of conventional CD4s and CD8s, they express the alpha-beta T cell receptor, which have a low affinity for their cognate ligands, which tend to be linearized peptides expressed on MHC. And in the case of gamma delta T cells, these are the gamma delta T cell receptors encoded on completely separate genetic loci that Alice was talking about when profiling the V regions. And these also have a low affinity for their cognate ligands, but like the antibodies, they tend to recognize their ligands directly without any processing in their native conformation. So as I mentioned, the alpha-beta T cell receptors tend to have this kind of stereotypical binding geometry where they dock directly to the peptide MHC, whereas the gamma delta T cell receptor has a much more flexible, diverse ligand binding geometry. It can bind just through the delta chain or just through the gamma chain. Sometimes the binding is CDR3 independent, sometimes it is CDR3 dependent. And while we only know about 40 ligands for gamma delta T cells across mouse and human, there is a concentration among them for what can be classified as stress ligands. So these are endogenous proteins which are either upregulated or somehow conformationally changed when a cell undergoes cellular stress. That could be UV irradiation, hypoxia, nutrient deprivation, and so on. Now, borrowing one of the figures from one of Bruno's many great reviews, just to showcase that gamma delta T cells can really be thought of as the Swiss Army knife within the tumor microenvironment. There's obviously the TCR-mediated binding to the cancer cells, but they can also mediate binding through the NKRs, as he brilliantly told us about in his talk. But they can also affect other immune cells in the TME. They can induce class switching. They can act as APCs for alpha-beta T cells. And while there are some documented deleterious effects, for the most part, they tend to benefit the patient. So when I was here in grad school, we were all very excited when Asha Lizadeh's paper came out in Nature Medicine, where they profiled by meta-analysis microarray data from tumor data that was deposited publicly. And what they saw is that when you look at a pan-cancer, that gamma delta T cells were the most favorable cellular correlate of protection across many different cancer types. And then more recently, there's been a great surge of exciting papers in our field. Can't really mention all of them due to time limitations, but there was a paper that came out a year ago from the Dutch Cancer Center that showed that it is the V delta 1s in the tumor microenvironment in patients that are class 1 deficient that respond to immune checkpoint blockade and protect the patients. And there's also been a great many papers coming out in the mouse models profiling specific V regions that are protective or not. Now, the reason to focus on gamma deltas in the cancer setting is that because they're not MHC restricted, they do not cause GVHD, which has been shown very clearly by the previous talks. And because of that, they have great potential for being non-self, off-the-shelf allogeneic treatments. And also, because of their broad anti-tumor specificity through the TCRs and the NKRs and other receptors, they tend to recognize ligands that aren't just patient-specific. But the limitations is that, as I mentioned, we only know of a few ligands, which make it hard sometimes to target them rationally. We have an incomplete understanding of gamma delta T cell function, and they are limited in circulation, although they're much more frequent in the mucosa. Now, the subset that I've been focusing on in my postdoc are the V gamma 9 V delta 2s. They're the most predominant subset in human circulation. They have broad anti-tumor specificity through a class of proteins called BTN2A1 and 3A1, which are part of a larger protein family called butyrophilins. So in the previous talks, when Alice and others were talking about phosphoantigen specificity, this is how that specificity is mediated. So when a cancer cell has a hyperactive mevalonate pathway, which is a very common hallmark of cancer cells due to their high proliferative needs and high need for cholesterol, the mevalonate pathway is in overdrive, and these metabolites labeled in blue, which we call phosphoantigens, bind to the intracellular portion of BTN3A1, causing a conformational change that is still being worked out that then allows BTN2A1 to be bound directly by the invariant portion of V gamma 9. So just to repeat that, because the naming is way too similar for comfort, BTN3A1 is the metabolic sensor, and 2A1 is the cognate ligand. And these proteins are expressed on healthy cells, but in an inactive conformation. So I wanted to understand what other cancer factors affect the detection of cancer cells by this subset of human gamma delta T cells. So I'm just going to be able to hit on the most salient points, but if you're interested in a much more detailed explanation, we published this recently in September, so I would invite you to read it in more detail. So we set it up by expanding V gamma 9 V delta 2 T cells with zeledronate from healthy donors. And then in parallel, we took Dowdy's, which are a B-cell lymphoma line that we knew is quite sensitive to killing by this subset of T cells. We generated a genome-wide knockout library of the Dowdy's using CRISPR. We primed them for killing even further by treating them with zeledronate. And then we co-cultured the gamma deltas with the Dowdy's for 24 hours. And then extracted the surviving fraction of the Dowdy's to identify which knockouts were protected from killing and also which knockouts were killed even more than the average. So what I'm showing you here on the upper right are the knockouts that were protected from killing. We knew right away that the screen worked relatively well because at the very top of the protected knockouts were the BTN3A1 and 2A1 knockouts themselves. We identified a slew of transcription factors that were unknown to be involved in this interaction. And then we were also surprised to find a very strong enrichment of subunits of the electron transport chain driving oxidative phosphorylation among the knockouts that were preferentially killed. At first, we thought this was simply a fitness defect and kind of ignored it. And then in a later screen, we realized there was much more to it than that. So in the follow-up screen, a much easier screen, we simply asked which genes regulate the surface expression of BTN3A. So again, we generated a genome-wide knockout library of the Dowdy's and simply sorted on BTN3A high and BTN3A low cells, and then compared the distribution of the guide RNA to understand which knockouts are enriched in each of the fractions. And just as a caveat, BTN3A 1, 2, and 3 are distinct genes encoding for distinct proteins, but they have pretty much identical ectodomains. So in principle, I'm looking at the sum total expression of these three proteins. Okay, so in the upper right, I'm showing you the knockouts that reduced expression of BTN3A, so these are positive regulators. And on the bottom left are the knockouts that increased expression of BTN3A, so these are negative regulators. Again, it passed all the quality control because the BTN3A genes themselves were at the top of the list. We identified the same set of transcription factors. About 75% of the hits, the ones labeled in orange, were concordant between the two screens, meaning the same hit either upregulated killing and BTN3 expression or downregulated killing and BTN3 expression, as you would expect logically. And then again, we saw this enrichment of various metabolic pathways among our significant hits. And in this case, fitness defects don't matter because everything's internally controlled for the cell numbers. So we delved deeper into the more granularly into the data of our metabolic hits to try and figure out if these pathways were coming up just due to a couple of enzymes that were hits or because we were actually picking up on the entire biosynthetic pathway. So just as examples, I'm showing you purine biosynthesis and glycan biosynthesis. And what we did here is we took each enzyme and we split it into two and applied a log-fold change if it was a significant hit. And if it's the same color, it means that this hit was concordant between the two screens. And what you can hopefully appreciate is that in the case of purine biosynthesis, pretty much every enzyme was a significant hit in our two screens, meaning that the entire pathway and probably its output is what matters here. So then we generated single gene knockouts in the DAUDIs to actually confirm what we saw, since these screens are generally just hypothesis generators and you really have to validate them on a single gene level to see if what you predict from the screen is true. We were able to confirm that a number of our hits caused significant either upregulation or downregulation of the BTN3A expression on the cell surface of the DAUDIs. And then we performed a very simple qPCR experiment, at first just to confirm that the transcription factors that we identified, such as IRF1 or RUNX1 and ZNF217, were actually acting on a transcription level and in the direction that we would predict from their role from the literature. And we did confirm that, but then we were surprised to find that when we looked in our metabolic knockouts, so when you knock out NDUF-A2, which is one of the many subunits of complex 1 in the electron transport chain, or when you knock out PPAT, which is the rate-limiting enzyme of purine biosynthesis, you also lead to transcriptional upregulation of the butyrophilins, which was surprising and indicated to us that there was some sort of metabolic sensing occurring that was being translated to the transcriptional level of the butyrophilins. So at first we confirmed that what we're seeing was real orthogonally with standard mitochondrial poisons. So when you block various complexes of the electron transport chain, you phenocopy the upregulation of BTN3A, although there were variable results with complex 1 inhibition, but a lot really robust results by blocking complex 5 or ATP synthase, complex 3, or just decoupling the whole ETC from ATP production with FCCP. You get this robust upregulation of the butyrophilins. And then we decided to try and figure out what was mediating the sensing. So there are a number of energy sensors that we tried. And basically what we happened upon is that when you turned on AMPK, which is the canonical energy sensor expressed in all cells, which senses when a cell is in an energy crisis by detecting the ratio of AMP to ATP, that when you turn on AMPK, either with an indirect AMPK agonist called ICAR, or with a direct agonist called compound 991, you again upregulate BTN3A on the DAUDIs. And then we collaborated with Jorgen Kubal and Joel Sebastian's labs in Utrecht University in the Netherlands. And with the help of their grad student, Angelo, he treated the patient-derived tumor organoids, specifically breast cancer organoids, with these agonists and showed that in their hands, these primary tumor organoids also upregulate BTN3A following AMPK activation. We were then excited to see that not only was BTN3A upregulated, but BTN2A1, the actual cognate ligand necessary for TCR activation, was also upregulated following AMPK activation. So what I'm showing you here are control knockouts, AVS1, so this is just a safe harbor cut, versus BTN3A knockout or BTN2A1 knockout. So you can see there's pretty much no staining with BTN2A1 knockout. BTN3A1, which is not the TCR, which is not the TCR ligand, knocking it out does not change the upregulation of BTN2A1. And then in the control knockouts, we see this upregulation. So that was quite exciting for us because now we realize that there would actually probably be a functional consequence as far as killing, since 2A1 is the actual ligand necessary for TCR engagement. So using an IncuCyte machine, which allows you in an incubator to follow your cell killing assay every couple of hours by reading out the GFP expressed in this case on the Dowdies, we cultured the gamma delta T cells with the Dowdies that were either pre-treated with the AMPK agonist or untreated. And what you can see here is that the pre-treated Dowdies were killed at a much faster rate compared to the untreated Dowdies. And then going back to our collaborators in the Netherlands, for over 10 years now, they have been working and pushing into clinical trials this cell therapy approach called TEG cells. So these are alpha-beta T cells that are engineered retrovirally to express defined V-gamma-9, V-delta-2 TCR clones that they have identified as potent killers. And in previous work, they have determined specific patient tumoroids that were either sensitive or insensitive to killing by these TEG cells. What we were excited to find is that when these tumoroids were pre-treated with the AMPK agonists, they had increased killing by the TEG cells. And what was even more exciting is that in the case of 34T, which they knew from previous work to be completely insensitive to killing by these TEG cells, the treatment with the AMPK agonists made them as good of a target for killing as the previously sensitive targets. So just to summarize what we showed here, so this was what was previously known about this system. There was only one known transcriptional transactivator known to be driving expression of butyrophilins, NLRC5. And then, as I described earlier, we knew what's happening at the cell surface. So what we added to this picture in our work is that when a cancer cell undergoes various metabolic stresses, which are quite common in cancer cells, especially in the stressful tumor microenvironment, this can lead to an energy crisis that would turn on AMPK, and then the AMPK would transcriptionally upregulate all of the components of the butyrophilin complex required for V-gamma-9, V-delta-2 TCR activation, that these same genes are also regulated on a transcriptional level by other transcription factors, which I didn't have time to show you. We mapped with cut and run a specific genomic loci where IRF1 and ZNF217 sit in the promoter regions regulating expression of these butyrophilins. There appears to be quite some important post-translational modifications going on in the ER and the Golgi, specifically N-link glycosylation and silylation that are, we haven't definitively shown it, but there's some data, preliminary data showing that it probably modulates the interaction with the TCR and the TCR affinity to the butyrophilins. There are quality control steps that make sure that the butyrophilin complex is fully assembled before it traffics up to the cell membrane. And then once it traffics up to the cell membrane, it can perform the metabolic stress sensing that was previously described. So as far as the fundamental implications, it was already known that the activation of this butyrophilin complex is metabolically dependent, but now we added to that by showing that the surface abundance is also metabolically dependent. Using functional genomics and CRISPR screening, we were able to uncover this mechanism by which gamma delta T cells surveil cellular homeostasis and detect cancer cells with aberrant metabolic profiles, which adds further evidence to this overarching theory that has been developing in the gamma delta field that gamma delta T cells are the stress sensing component of the immune system. And then as far as therapeutic implications, as previous talks have mentioned, there's now a very rich diversity of approaches that are being developed in biotech as well as academia, trying to utilize the power of gamma delta T cells in the context of cancer. There's the adoptive cell therapy approaches of gamma delta CARs, as well as using defined gamma delta TCR clones. There's bispecific engagers. And in all of these cases, having more ligand on the target cell would improve, in theory, it should improve the efficacy of these approaches. So perhaps there's a way in which that could be synergized, and the AMPK agonist could be paired with V-gamma-9, V-delta-2 immunotherapies. So I'd like to thank people in our lab who have been instrumental for this project, as well as Aaron Adams at the University of Chicago, who provided some key unique reagents that made the work possible, the collaborators in the Netherlands that pushed this forward, and my funding sources. Thank you.
Murad from Decode. I have a great presentation.
Thank you.
I have a question about the AMPK agonist theory that if you could combine it with other therapies.
Yeah.
Do you see that increase in healthy cells as well, or is it something specific to cancer cells that BTN3A would increase on them? So I'm coming at it from the perspective of toxicity you would see with that combination.
Right, that makes sense. We still have to test that more thoroughly. I would expect it also to increase on healthy cells, but the beauty of the system is that it's a two-signal system. So simply having more butyrophilin on the cell surface will never engage the TCR on its own. It has to be combined with this aberrant mevalonate pathway because in a healthy cell, it's an inactive conformation that is never accessible to the TCR, which is one of the main reasons why there's a great safety profile for these cells. So without the aberrant mevalonate pathway, just having more of it on the surface should not in principle cause toxicity.
Great, thanks.
Yeah. Great. Thank you.